Animal cloning. I just remember the quote by a French journalist, Claude Orelin, stating, "Cloning will enable mankind to reach eternal life," which tells the importance of the cloning techniques. What is meant by cloning? Cloning is a process of generating or producing a genetically identical copy of a cell or a gene or an organism. Clones are the organisms that are the genetic exact copies, where every single bit of their DNA are identical. The naturally produced clones are the identical twins. They share the exact copies of genes. Are there types under the cloning techniques? Yes, there are three types of cloning: gene cloning, therapeutic cloning, and reproductive cloning, also called as animal cloning. Gene cloning is the process of isolating the gene of interest from a desired organism to produce the identical copies of it. In 1973, Stanley Cohen and Herbert Boyer developed the process to produce recombinant DNA, which is a form of artificial DNA. Therapeutic cloning uses the cloned embryos to extract the stem cells from it without placing them in the womb. We know that the stem cells are totipotent, that is, capable of differentiating into 200 cell types in the human body. Reproductive cloning is the process of implanting the cloned embryo into the womb of an organism. The embryo later develops into the offspring, identical to the animal from which it has been derived. For every big invention, there are always some root cause which makes the technique or the invention to be defined or shaped, and many people are there behind those inventions. Now we are going back to the history from where the ideas of cloning came and the people who worked for it. In 1885, the first ever demonstration of artificial embryo twinning was conducted by a German biologist, Hans Adolf Edward Reich, in sea urchin. He showed that it was possible to separate the cells by shaking the two-celled sea urchin embryos, and once separated, each cell developed into a complete sea urchin. This experiment showed that each cell in the early embryo has its own set of genetic instructions and it can grow into a full organism. In 1902, a German embryologist, Hans Perman, conducted artificial embryo twinning technique in salamander. Sperman fashioned a tiny noose from a strand of baby hair and tightened it between two cells of a salamander embryo until they got separated. Each cell grew into an adult salamander. Sperman also tried to divide more advanced salamander embryos using this method, but he found that cells from these embryos were not as successful at developing into adult salamanders. In 1928, Hans Perman conducted an experiment which showed the control of embryonic development by the nucleus from the early embryonic cells that directs the complete growth of a salamander, effectively substituting for the nucleus in a fertilized egg. In 1952, the research scientists Robert Briggs and Thomas King created many normal tadpole clones using the nucleus from the early embryos as like Sperman's experiment. But only a few tadpole clones survived but grew abnormally. In 1958, John Gurdon, a British biologist, transplanted the nucleus of the intestinal cells from the tadpole into the enucleated frog egg and produced tadpoles genetically identical to the tadpole which donated the intestinal egg. This experiment showed that nuclei from somatic cells in a fully developed animal could be used for cloning. That is, the cells could retain all the genetic material even as they divide and differentiate. In 1975, Dr. J. Derek Bromhull transferred a nucleus from a rabbit embryo cell into the enucleated egg cell of a rabbit and considered it a success after seeing the development of an advanced embryo after a couple of days. But he never transplanted the developed embryo into the surrogate rabbit to see the further development. In 1996, Ian Wilmot and Kate Campbell used the donor nuclei from the cultured laboratory sheep cells and transferred them into the enucleated sheep egg cell. Two lambs were developed and named as Megan and Morak, and in the same year, both the scientists produced a lamb named Dolly by transferring the nucleus from an adult sheep's udder cell into an enucleated egg. This experiment is a remarkable one in the history of cloning. In 1997, Don Wolf and his research team produced the first primate by embryonic cell nuclear transfer. After Dolly, between 1998 and 1999, scientists wanted to clone some other mammalian species, and hence they produced many animal clones by cloning techniques. In 2001, scientists thought to produce the endangered and extinct species. They decided to use the closely related animal to serve as egg donors and surrogates. Thus, scientists have chosen gourd and mouflon as they are relatively close to cattle and sheep. In 2007, Shokret Mitalipov and a group of scientists transferred an adult monkey cell nucleus into the enucleated egg cell. This embryo was allowed to develop for some time, and the cells were grown in the Petri plate, which constitutes the embryonic stem cells. Thus, they proved nuclear transfer in primate is possible, which scientists tried for many years. In 2013, the same team fused the skin cell and donated an egg cell from a patient. They modified the culture medium and a series of electrical impulses that are used to stimulate the dividing of egg. This enabled them to produce human embryonic stem cells with somatic cell nuclear transfer. The first study of cloning by Hans Adolf Edward Reich in salamander made a breakthrough for other researchers in further findings in cloning. And the findings of John Gurdon in frog have inspired Ian Wilmot in the development of cloned sheep named Dolly, which is a very popular one. Ian Wilmot said that the birth of Dolly and the new understanding of the opportunity to change the functioning of cells made researchers consider other possible ways of modifying cells. Thus, many scientists struggled a lot to develop the cloning techniques, and it became a medical boon in many developed countries where the developing countries are still trying to produce the cloned animals where the facilities are inadequate. Artificial embryo twinning is a technique similar to the natural process which creates the identical twins. We know that in nature, identical twins are produced when the early embryo splits into two and becomes separate individuals with exact copies of gene replicas. Whereas artificial twinning also follows the same approach, but can be carried out in petri dish instead of in the womb of an organism. As all those embryos are derived from the same fertilized egg, they are genetically identical. Through embryo splitting, several embryos with identical genetic features are obtained. Embryo splitting has potentially been helpful for gaining a higher number of embryos from one dividing embryo. Hence, these procedures are commonly and successfully applied in livestock breeding. In livestock, for example, a cow with desired traits is selected and treated with hormones so that the cow could superovulate and produce many eggs at one time. The fertilized egg is the early embryo and it will be collected in the petri plate. The embryo is splitted artificially by a fine razor blade into smaller early embryos and are grown in a laboratory for a few days. The healthy embryo will be selected and transferred into the surrogate mother. In the case of some animals like pigs, they naturally produce many offspring in each pregnancy. The implantation of one embryo may not be recognized by its own body and could reject it. Somatic nuclear transfer. Somatic cell nuclear transfer, which is also known as nuclear transfer, that differs from artificial embryo twinning but produces the same result, that is, clones with exact genetic material. Somatic cell nuclear transfer is a technique where the nucleus of somatic cell, that is the body cell, is transferred to the cytoplasm of an enucleated egg. The enucleated egg that has had its own nucleus removed. The enucleated egg and the somatic cell nucleus is fused together with shock and will begin to divide and become an organism with almost identical DNA to the donor organism which donated the somatic nucleus. In 1996, Ian Wilmot and Keith Campbell created the first mammal with somatic cell nuclear transfer. Ian Wilmot and Campbell produced a lamb by transferring udder cell nucleus from an adult sheep into an enucleated egg. This is the first time ever a mammal had been cloned from an adult somatic cell. There are several breeds of sheep in the world, but in the case of Dolly, scientists selected Scottish blackface sheep, which has black face, and Finn Dorset sheep, which is all of white. The steps to produce Dolly involved many procedures. First, the egg cell of black-faced sheep was taken. Meanwhile, the mammary cell from the Finn Dorset was removed. Then the cytoplasm of black-faced sheep and the somatic nucleus of Finn Dorset were fused together with shock and the embryo was developed. Later, the embryo was placed in the uterus of the surrogate mother and Dolly was born. Dolly genetically resembled Finn Dorset mammary cells and not the black face, which clearly demonstrated that she was a successful clone. Of 277 attempts, only one produced an embryo that was carried by a surrogate, and after 148 days, Dolly was born on 5th July 1996 with normal gestation. Dolly remained healthy with well-developed body organs, which are derived genetically from nuclear DNA. On February 14, 2003, Dolly was found dead, suffering from progressive lung disease. Now her body was preserved and displayed at the National Museum of Scotland in Edinburgh. The arrival of Dolly brought cloning into the light that started the conversations about further implications of cloning and the thoughts about human cloning and the stem cell research into the public. Advantages: elite, high-value animals can be cloned in large numbers. Endangered animals can be produced through cloning to preserve the species. Sparse stem cells can be produced. Pets can be cloned. Genetically modified animals can be quickly reproduced. Animals can be cloned for xenotransplantation. Disadvantages: animals produced by cloning may suffer bad health. Absence of genetic variation. The cloned animals may be unable to adapt to changing environments and evolve. Cloning success rate is very poor. Cloning is expensive and labor intensive. Applications: animals are produced for the production of drugs. Pharmaceutical production of medically important molecules in milk, example, human antithrombin 3, to regulate blood clotting while surgery, and many other compounds can be produced. Cloned animals can be used as animal models for laboratory experiments. The endangered and the organisms on the verge of extinction can be produced through cloning in order to conserve and protect biodiversity. Transgenic applications. The cultured cells can be genetically modified with desired character in the laboratory, so that the transgenic animal with desired character is produced. Those modified cells can be used in nuclear transfer. Human cell based therapy. In order to replace the affected tissue with the cloned cells and restore its specific function, which is similar to the gene therapy. Done in conditions like diabetes, muscular dystrophy, cancers, various neurological disorders, spinal cord injury, etc.